Okay, well, welcome everyone. My name is Samuel Clark. I'm the Director of R&D at Precision Nanosystems. I'm here with my team members today to present some of our findings relating to the development of a model mRNA therapeutic. So this slide gives a brief overview of the presentation today, some of the key aspects that I'd like to cover. What I'm going to start with is a bit of an introduction to Precision Nanosystems and our core technologies. And then I'd like to present some scientific data demonstrating the use of our NxGen microfluidics for manufacturing lipid nanoparticles, as well as demonstrate how these technologies can be used to produce a mRNA drug. Okay, so I wanted to start with the vision for Precision Nanosystems. Our vision is to accelerate the creation of transformative medicine that significantly impacts human well-being. This slide describes some of the main application areas where we work and where a lot of our clients work. We work extensively in the areas of new vaccines, including genetic vaccines. We work in the area of cell therapy and regenerative medicine, as well as immuno-oncology and targeted therapeutics, small molecule delivery, RNA and DNA therapeutics, and gene editing. So as a company, we're really working at the forefront of transformative medicines and nanomedicines. Our nanoassembler platform for manufacturing nanoparticles has been deployed worldwide. We now have over 350 instruments placed. We work with many different academic groups, including over 90 groups. We work with over 100 industry groups, including biotech and 19 of the 25 top pharma. And finally, our platform has been featured in more than 150 peer-reviewed publications, really showcasing the breadth of application and adoption of our technologies in this new area of drug development. And so what I would like to do before I get into some of the scientific data is to give an overview of some of the core technologies that we have at Precision Nanosystems, and then just to help set up some of the scientific data. So we really have two core technologies that I'd like to highlight. The first one, shown on the left-hand side, is our nanoparticle technology. At Precision Nanosystems, we have developed proprietary formulations of nanoparticles that can be used for a range of applications, including gene delivery. One of these technologies is called GenVoy ionizable lipid mix, and I'll provide some more details of that as we go through the presentation. The second core technology that I would like to highlight is our manufacturing technology for the manufacture of nanoparticles. We have a microfluidic-based approach for highly scalable production of high-quality nanoparticles, and this technology has been implemented on our nanoassembler instrumentation platform. And again, I'll go through the details of this technology and provide you with some data on how it works. So I'd like to start with our nanoparticle technology and specifically talk about GenVoy ILM lipid nanoparticles. For those unfamiliar with lipid nanoparticles, this slide provides a bit of an overview of this technology and some of the advantages. Lipid nanoparticles are a very effective tool for gene delivery and very effective as delivery vehicles. Recently, lipid nanoparticles as a technology have been clinically approved with the approval of Onpattro or patisiran. This is a siRNA-based drug for the treatment of liver disease. So the advantages of lipid nanoparticles for these applications include that they protect the payload from degradation. These lipid nanoparticles allow for efficient delivery of the nucleic acid into primary cells. In vivo, they typically traffic to the liver, and as such have high utility in the treatment of liver disease, as well as applications where the liver is used to produce therapeutic proteins. These lipid nanoparticles are designed to facilitate cellular uptake, and they're also designed once in cells to release the nucleic acid into the cytoplasm. So overall, lipid nanoparticles are a very elegant approach by design to deliver nucleic acids to cells. This slide describes our GenVoy ILM technology, which is a lipid nanoparticle formulation designed for gene delivery applications. So the graph on the left-hand side is an example of how you can use GenVoy ILM for gene knockdown. In this particular example, GenVoy ILM was used to encapsulate an siRNA encoding for factor VII. These lipid nanoparticles were administered into mice, and the results in the graph show significant knockdown of factor VII from serum. Similarly, on the right-hand side is an example of the use of GenVoy ILM for the delivery of a messenger RNA. This is for gene expression in vivo. In this example, messenger RNA encoding for luciferase was encapsulated within the lipid nanoparticles and administered to mice, and the results there showing high expression of the luciferase reporter. So overall, GenVoy ILM is really a powerful formulation for gene delivery applications. Now I'd like to introduce our second core technology, which is our NxGen microfluidics platform. And this is a platform that allows for the scalable manufacture of lipid nanoparticles. And so just to start off here, the point I wanted to make with this slide is that nanoparticles are much more complex than previous generations of drugs. So this example here shows a RNA lipid nanoparticle, which is about 100 nanometers in size and contains about 100,000 molecules. And so the complexity of these lipid nanoparticles is much greater than previous generations of drugs, such as antibodies or proteins or small molecules. So with this complexity, one of the key challenges with nanoparticles is their manufacturing. In particular, an aspect of the production of these type of nanoparticles that is really important is the self-assembly process. This slide here and this vignette, this schematic, shows an example of the self-assembly of lipids along with nucleic acid in a process that ultimately leads to the formation of the nanoparticles. So the generation of high quality reproducible nanoparticles is really a function of how well the self-assembly process can be controlled during the manufacturing. And so the approach we take at Precision Nanosystems is to use microfluidics technology to carefully control the self-assembly and the mixing conditions for nanoparticle production. And in this slide, this shows a schematic of the microfluidic concept. In this particular example, we have a case where lipids and ethanol are combined with nucleic acids in an aqueous solution under controlled flow conditions and then run through a microfluidic mixer for very rapid mixing. So this approach allows us to make very high quality nanoparticles in a very reproducible way and in a way that is easily scalable. And so this slide provides an overview of different manufacturing techniques and the evolution of manufacturing techniques for nanoparticles. Initial techniques in the field were largely based on high energy techniques. These included techniques like sonication and extrusion. These techniques had challenges with limited applications and difficulty to reproduce the synthesis batch to batch, as well as difficulty to scale. Improvements were made in terms of approaches that rely on inline macro mixing. An example here is the use of T-tubes. These techniques did solve some problems of the high energy techniques, but also have limited applications and still have challenges with reproducibility. And so it's really over the last 10 years now that microfluidic approaches have really helped to solve some key challenges in the area of manufacturing nanoparticles. One example here is the staggered herringbone microfluidic mixer technology, which is now widely published and has been utilized in many of Precision Nanosystems microfluidic manufacturing technologies and products. these technologies have really solved issues of reproducibility, allowing for the production of nanoparticles of very high quality and in a very consistent way, batch to batch. One of the remaining challenges with traditional microfluidics approaches is that scaling up, particularly to very large volumes, has remained somewhat of a challenge. And so what I'll focus on today is our new NxGen microfluidics technology, which we feel is really poised to revolutionize the approach to manufacturing of nanoparticles. One of the key advantages that this technology brings is the ability to easily scale production from very small volumes all the way to very large volumes in a very fast, simple manner, while retaining all the advantages of microfluidics, including the reproducibility and the high quality nature of the nanoparticles produced. And so this next generation microfluidics, they're precise, scalable, and what we'll do in the remainder of the talk is really showcase some of these advantages through our data. The final slide here in this section, I just wanted to highlight that these NxGen microfluidics have now been implemented across an instrumentation platform. And this instrumentation platform collectively supports all stages in the discovery and development, the preclinical development and the clinical development of nanoparticle-based drugs. The microfluidic technology, along with the instrumentation, allows for the production of nanoparticles in the microliter volumes, all the way up to tens of liters or more than 20 liters per hour of throughput. And so now I'd like to shift and move into presenting some results of a scientific study that we performed here at Precision Nanosystems to demonstrate how mRNA-based drugs can be rapidly developed using both the NxGen microfluidics as well as our GenVoy ILM nanoparticle reagent technology. So this slide provides some background on the model therapeutic approach that we selected. We selected an mRNA encoding for erythropoietin to encapsulate within lipid nanoparticles and to use as a model drug. Just as a little bit of background here, anemia is caused by kidney disease or cancer chemotherapy, and the treatment of anemia is with a recombinant erythropoietin. And so as shown on the slide here, the treatment usually involves the administration of erythropoietin protein directly to the patient. Anemic patients have a low red blood cell count, and so the administration of EPO leads to increased production of red blood cells and to ultimately correction of anemia. And so our approach with the lipid nanoparticle encapsulating mRNA encoding erythropoietin was to develop this nanoparticle and administer it into mice to demonstrate how we can use the liver to produce erythropoietin and to have a therapeutic effect that is similar to that observed by administering the protein directly. So this is the model system that we've used, and as we go through the data, we'll be showing biological activity that relates to this therapeutic output. And so for the first set of data, where we started with this work was on the small scale using the Ignite instrument. And our initial goal was to compare the staggered herringbone mixer along with our NxGen microfluidics technology with the objective to show that these two mixers at small scale generate equivalent nanoparticles with very high quality properties. And so here's our first set of data based on this system and this comparison. The specific details of the experiment are described on the right-hand side in the table. As I've mentioned, we're using the GenVoy ILM formulation. We're using an mRNA encoding for erythropoietin, EPO. We're using the process parameters described in the table to generate the nanoparticles on the instrumentation, following that with downstream dilution and ultrafiltration for purification. And so these two graphs show the analytical characterization of these nanoparticles. On the left-hand side, the graph shows size and polydispersity. You can see is that the nanoparticles are 75 nanometers in size and have a polydispersity of less than 0.1. So these are very high quality nanoparticles compared to conventional techniques. And in our direct comparison of the staggered herringbone mixer, as well as the NxGen microfluidics, we can see that the characteristics of these particles are equivalent regardless of mixer. Similarly, on the right-hand side, we compared mixers and the encapsulation efficiency of the mRNA. In both cases, encapsulation efficiency is over 90%, so it's a very efficient and effective encapsulation process. So now we took this a step further by examining the biological activity of these mRNA lipid nanoparticles encoding for erythropoietin. The approach we took was to administer these lipid nanoparticles into mice and to assess the erythropoietin levels in serum following this administration. And so the graph on the left-hand side shows total serum erythropoietin for the different conditions, and also at the doses indicated. And so this result shows equivalent production of erythropoietin with the nanoparticles produced on both the Ignite Classic employing staggered herringbone mixer technology and the Ignite with the NxGen microfluidics technology. We took this a step further now, and we looked for a therapeutic effect of these lipid nanoparticles in mice by assessing the hematocrit, which is a measure of the red blood cell production. And so this data on the right-hand side shows that, as the doses indicated, that there is an increase in the hematocrit over controls from about 50 to 60%, and that this increase is equivalent for both of the mixers evaluated here. So overall, this data gives us a lot of confidence that at small scale, we can produce very high quality nanoparticles with the NxGen microfluidics technology, and that these nanoparticles are equivalent to those produced on the classic staggered herringbone mixer. So this data set provides us with a very strong baseline now to go and scale up this process and drug product. And so in the final section here, what I'll do is highlight the scientific data that we have demonstrating this. And this is really where one of the key advantages of the NxGen microfluidics comes in particular, our ability to rapidly scale up these type of drug products. So before I get to the data, what I would like to highlight is our philosophy and our approach on developing these type of drugs. The approach we take in general is to consider the different unit operations involved in producing a drug product. This includes the microfluidic mixing step. It includes inline dilution, as well as downstream steps, such as tangential flow filtration and sterile filtration, which collectively lead to the final drug product used to treat disease. So this is the same approach that we used in the data that I showed previously using the Ignite system. And this is the approach now that we're going to apply to scale up this drug product using additional instrumentation. So here are more of the specific details of our experimental design. What we've done is we've scaled up the reactions. We've scaled up the batch sizes on the Ignite system to 4 milliliters from 1.5 in the previous data set. And then we will utilize the Blaze system to produce larger volumes of these nanoparticles, up to 55 milliliters, and the GMP system to produce an even larger volume at 325 milliliters. So all of this study employs the NxGen mixer. The table here provides a full list of the parameters. One important point to highlight is the total flow rate, where on Ignite it's 12 mils per minute, And as we move to Blaze and then to GMP, we are able to progressively increase the flow rates up to 200 mils per minute. And this is one of the main advantages in that we can create a much faster, a much larger production rate for the production of the nanoparticles. All samples were inline diluted in the data that I'm going to show, unless indicated otherwise. And we apply downstream processing, either ultrafiltration or tangential flow filtration, or both of these technologies. And so the first data set I'll start with is the analytical characterization of these mRNA lipid nanoparticles that have been produced at different batch sizes using the NxGen technology. And so the graph on the left-hand side is size and polydispersity. And the samples indicated describe the instrument used to produce the nanoparticle. They describe the flow rate applied to produce these nanoparticles, and provide an indication of whether or not inline dilution is used. And so in terms of size, we can see that the size of the nanoparticles across all of these different conditions are equivalent, and polydispersity is very low, in all cases below 0.1. Similarly, when we look to encapsulation efficiency across these range of conditions, we can see that encapsulation efficiency is equivalent regardless of batch size and regardless of other conditions tested. And so overall, this data set really strongly supports the fact that these lipid nanoparticles produced at different batch sizes using our instruments and the NxGen microfluidics both have very high quality properties and equivalent properties irrespective of scale. And so we went on to assess the biological activity of these different samples produced at different scales. And so the graph on the left-hand side shows the production of erythropoietin in serum at the doses indicated. And what the graph shows for the different samples, reflecting different batch sizes and operating conditions, is that the amount of erythropoietin in the serum is equivalent. And so in these cases, we're comparing batch sizes of about 4 milliliters on the Ignite system to over 325 milliliters on the GMP system, using flow rates ranging from 12 mils per minute all the way to 200 mils per minute. So again, I just wanted to emphasize the biological activity shown by erythropoietin levels in serum equivalent across all these conditions. We further compared the effect of downstream processing by looking at and comparing ultrafiltration and tangential flow filtration. And these are indicated in the groupings on the graph on the left-hand side. And again, can conclude that the approach for downstream processing does not impact the level of EPO in serum. So we also, again, looked to measure the therapeutic effect of these different lipid nanoparticles by measuring the increase in red blood cell production through an increase in the percent hematocrit. And this is the graph on the right-hand side. Again, what we can see at the doses indicated is equivalent increase in hematocrit from about 50% to over 60% in all cases, irrespective of scale. Similarly, there was no observed effect or difference in the downstream processing technique when we compared ultrafiltration and tangential flow filtration. So overall, this data set, we feel really strongly supports the fact that the NxGen microfluidics technology, it allows for very simplified and easy scale up, and in addition, allows for the production of very reproducible and high quality nanoparticles. And so I just wanted to come back and conclude with again, our philosophy on generating and developing lipid nanoparticle-based drugs. Our philosophy is to think about the different unit steps involved in the production of a nanoparticle, starting with microfluidic mixing, inline dilution, and then downstream steps, including tangential flow filtration and stereofiltration to produce the final drug product. This is an approach we take at small scale, as we've demonstrated on Ignite, and this is an approach that we have applied to scale up nanoparticles. Furthermore, it's an approach that's standard in GMP manufacturing, and so overall, we feel that it's a methodology that allows for a reduction in risk during transition from research to development, and can help accelerate the development of nanomedicine-based drugs. So with that, I would really like to acknowledge the research and development team at Precision Nanosystems. Here, pictured here on the slide, we have a really fantastic team, a really dedicated team here with a highly multidisciplinary background, and have really been key contributors in the development of these core technologies that I've highlighted. In addition, you can always reach out to Precision Nanosystems by the email here on the slide, as well as going to our website. So thank you.